light receptors, photoreceptors for A-level biology. So you want to be able to explain how the distribution of rods and cones and the connections they make in the optic nerve lead to differences in sensitivity and visual acuity. So just a quick recap of the structure of the eye. Around the back of the eye, shown here in like a red colour, that is the retina. And then there's a little triangular indent here, which is called the fovea. Got the blind spot, and then the optic nerve. And then at the front, you've got the lens, which can make you short-sighted or long-sighted, the pupil, and then the iris. So there are two types of photoreceptor, rods and cones. And rods are found in the peripheral parts of the retina, so around the outsides. Whereas cones are packed together in this tiny little triangular indent here called the fovea. So rods are found around the periphery, whereas cones are found packed in the fovea. Rods give information in black and white, so it's called monochromatic vision. Whereas cones give information in colour, so it's known as trichromatic vision. And there's three types of cones: red sensitive, green sensitive, and blue sensitive. You can't say red cones, green cones, blue cones, because they're not actually red, green and blue. They're just sensitive to that type of light. So you've got red sensitive cones, green sensitive cones, and blue sensitive cones. And they're stimulated in different proportions, so you can see different colours. So how do photoreceptors work? So light enters the eye, it hits the photoreceptor and is absorbed by light sensitive pigments, and light bleaches the pigments, causing a chemical change and altering the membrane permeability to sodium. And then a generator potential is created, and if this is enough to reach threshold, eventually the nerve impulse will be transmitted along the bipolar neuron, and then bipolar neurons connect to connect the photoreceptors to the optic nerve, which takes impulses to the brain. So rods, there are 180 molecules of light sensitive pigment called rhodopsin, and this breaks down when exposed to light into retinal and opsin, and it causes a transmitter substance to be released, which results in an impulse being sent down the optic nerve. And when you enter a dark room, the pigment rhodopsin is restored because retinal and opsin join back together to form rhodopsin, and that is called dark adaptation. So when you initially turn the light off, you can't see anything, but after a while you can gradually see more and more as you go into the dark. Then you've got cones. Cones have a pigment called iodopsin. You can see that they are cone shaped, shown here on the left. And the iodopsin is broken down by high light intensities. And it's broken down into photopsin and L-trans-retinine. And remember, the cones are packed tightly in their fovea. Now, in terms of the sensitivity, rods are very sensitive to light. They will fire action potentials even in dim light. And this is because many rods join one neuron. So that means that many weak generator potentials can combine to reach the threshold and trigger an action potential in the bipolar neuron, which will then be passed to the optic nerve. Whereas cones are less sensitive than rods, they only fire action potential in bright light, so you can only see colour when there's adequate light. And the reason is because one cone joins one bipolar neuron. So it takes more light to actually reach the threshold and trigger an action potential in the bipolar neuron. Visual acuity. So visual acuity is the ability to tell apart 2 points that are close together. Rods have quite low visual acuity. So when you're in the dark, you can only really see the outlines of objects. It's not clear detail. So rods have low visual acuity because many rods join to the same bipolar neuron. So light from two objects that are close together can't be told apart. The brain doesn't get separate information because it all gets amalgamated. Cones, on the other hand, have high visual acuity because cones are close together and one cone joins to one bipolar neuron. So when light from 2 points hits 2 cones, 2 separate action potentials are fired. So that means the brain can distinguish the separate information, and it can distinguish the 2 points that are close together as 2 separate points. So a little summary table here. Rods are mainly located in the peripheral parts of the retina, whereas cones are located in the fovea. Rods give information black and white, whereas cones give information in colour. For rods, many rods join one neuron. There's convergence. Whereas cones, one cone joins one individual neuron. Rods have high sensitivity to light, so they only need a little bit of light to stimulate them. Cones have low sensitivity to light, they work in bright light. Rods give low visual acuity, whereas cones give high visual acuity. So in terms of rods, there are 120 rods per million in the eye. 120 million rods per eye, whereas there's only 6 million cones per eye, so there's a lot less cones. In terms of retinal convergence, about 15 to 45 rods can converge to one bipolar cell, whereas cones in the fovea, one cone will join to one bipolar cell. In terms of sensitivity, rods have good sensitivity. One photon alone gives a response. Cones have poor sensitivity. You need several hundred photons to stimulate a cone. Rods have poor visual acuity, whereas cones have good visual acuity.